Holly Hunsberger is a postdoctoral research scientist with a dual appointment at Columbia University and the New York State Psychiatric Institute. She received her PhD from West Virginia University in the area of behavioral neuroscience. There, she studied glutamate's role in Alzheimer's disease pathology using novel microelectrode array technology. She is now in Dr. Christine Denny's laboratory, where she received a grant to study the sex-specific impact of anxiety on Alzheimer's disease progression. Additionally, she has a fellowship with the Columbia Technology Ventures Office, where she works on early stage technology assessments, writing marketing abstracts, and preparing marketing campaigns. Over to you, Holly.
Thank you. Thank you so much for the introduction. I will share my screen. Okay, does that look good?
Yeah.
Okay, perfect. I'm gonna minimize this video. Okay, great. So my name is Holly. Today I'm gonna be talking to you all about learning and memory. So I'm gonna try to give a big picture, a little bit of background on the history, and then kind of get into the work that we specifically do in our lab and really kind of show you some fun techniques that we use now in the field of learning and memory. Okay, so you guys are maybe familiar with the Pixar movie called Inside Out. So in this movie, viewers follow 5 emotions as they help kind of mold this young girl. And what's pretty interesting is how they represent memories in this movie. They actually do a very good job. So each memory is colored by emotion: fear, sadness, joy, disgust, and anger. And there are core memories that make up the main being of the girl. There are memories that get deleted. There are memories that move from short-term to long-term. And although this is kind of oversimplistic representation of memory, this is essentially what our lab is interested in. So how do memories form? How do they get stored? What makes some memories more important than others or salient? And what happens when you can't remember a certain memory? Is it lost? Is it there, or is it hidden somewhere in the brain? And so I think the broad topic is, what are memories made of? I think that's what we're all really interested in. And so the question is, does the brain store memories? So this is a very obvious theme in contemporary neuroscience, hotly debated all the time. And for some, it was obvious that memory is represented in the brain, that is physically. But others argued that it was stored in the mind more psychically. And so Richard Seaman, shown here on the left, argued not only that information is encoded into memory and that there are memory traces, what he defined as memory traces, he called them engrams, that conserve the changes. However, he also contended that changes in the brain are inherited, so that you could essentially pass down memories from one person to another. So his theory fell short, and it was largely disputed because of that. So then Carl Lashley came along, and he suggested that the memory trace could not exist in any specific part of the brain, but that it was widely distributed throughout. And so in Lashley's experiments, this occurred around 1929 to 1950. He would train rats in a maze, and he would remove tissue from different parts of the brain, and then he would put them back into the maze to see how their memory was affected. And increasingly, the amount of tissue that was removed ended up degrading the memory. But more remarkably, where the tissue was removed really made no difference. So then in 1953, patient HM came along, and he's probably one of the most famous in neuroscience that we study. There are many books written about him, Henry. And he was an American man. He ended up having a bilateral lobectomy. So that essentially means they just removed 2 chunks out of both sides of his brain in an attempt to cure his epilepsy. So he suffered from epilepsy, drugs weren't working, so they removed 2 parts of his brain. And this did cure his epilepsy, but he could no longer form memories. And he was actually studied up until he died in 2008. And so this really led scientists to start thinking, that there were certain parts of the brain that were more important for memory than others. And this was really important to discovering, the hippocampal area. And so the hippocampus is very important for memory, encoding, and retrieval, and we know that now. The name hippocampus is Greek. So hippos means horse, and campos means sea monster, because the hippocampus kind of looks like a seahorse. And this structure actually was discovered in the 1500s. They just didn't know what the functional role was until the 1950s. And its function is still being examined today. So the hippocampus here works as a circuit. So there are multiple subregions, as you can see, the DG, CA3, and CA1, and the EC, which is the entorhinal cortex. And they all feed into one another. So the DG loops and sends signals into the CA3, which sends signals into the CA1. And these all kind of work together to create memories, encode memories, and help you retrieve memories later on. And this is called the trisynaptic pathway. What I'm showing here is what this looks in a mouse brain. This image here is where your hippocampus is located in the human brain. And so within the hippocampus, there are cells that are important for memory encoding and retrieval. And these are called memory traces, or engrams, like I had mentioned before. Those terms still hold true today. And when I talk about memory traces, I first want to define everything before I start going into data and pictures of what all this looks like. So these are cells that are activated by learning. So for example, if you had to learn a new TikTok dance, these cells would turn on. These cells would also change permanently as a consequence of you learning the dance. And these cells would then turn back on when you heard the same song. So they're activated by learning, they have enduring cellular changes, and they can become reactivated when they see a similar context. And within that memory trace cell is a protein called ARC. So ARC is an immediate early gene, and all that means is that this gene becomes active or gets turned on after a learning experience. And this image is representative of the sparse expression of ARC in the regions of the hippocampus. So you remember I showed you the hippocampus before. It's the same structure here, just with the labeling of that ARC protein in the cell. So all of these purple dots are cells here. So you can see it's pretty sparse in this area. It's more dense in other areas, and that will become important later. And so learning induces ARC. At baseline levels, ARC expression is quite low. So if you look at the control, the yellow here is the ARC. Upon learning, you see this increase in ARC, that's the red. And then when you retrieve that same memory, you get some of those red cells that are activated again. So same thing of what I've been saying, just more images to show that. So the summary of learning and memory throughout the hippocampus, you learn your TikTok dance. This gets ARC activated, which then leads to signals throughout the hippocampus. So this is kind of a basic idea of encoding, activation, storage, and retrieval. So what has happened in the past 65 years since we first discovered the functions of the hippocampus? I would say in the last 5 to 10 years, we've made much progress on identifying and manipulating memories throughout the brain. So this is an image from a review back in 2015. I could probably update this now, just showing some of the main regions that labs have identified. So you'll see the hippocampus here, some in the amygdala, which is known for emotion regulation, and some are even shown in the prefrontal cortex. So they're found basically throughout the brain. So with the previous work implicating ARC and the hippocampus in memory formation, our main objective was to create a mouse line that had a genetic label that would last indefinitely. So we thought if you could tag a memory cell, like I mentioned before, that gets activated from learning with the ARC protein, then we could study how memories change with age, in disease, and where they go long term. So our lab actually created a mouse that tags ARC positive cells with a green fluorescent protein in the presence of a drug. So don't worry about what all these letters mean. This is just how we denote mouse lines, but essentially you inject this drug and those cells will turn green. So this is what it would look like in an actual brain staining. So you would tag a memory, that would be the green cells. You could also look at the retrieval cells, staining them a different color. And then you can look at the overlap of these two cells, meaning that both cells were important for forming that memory, and that's what we call a memory trace. So we now have the power to look at the physical cell that's important for shaping our memories. So I'll show you guys another example of this. So this is again the hippocampus that I've shown before. And the green here is the learning. So any cells that you see, any green dots here, that's a learning cell. That cell turned on when the mouse learned something. These red dots are all the remembering, the retrieval of what they learned. And the yellow is actually the overlap. So when the green and the red cell overlap, that's what we are calling a memory trace. So both of those cells turned on for the encoding and retrieval of that memory. So that right there is a physical representation of one of your memories. Okay, so now that we had a mouse model that would allow us to look at memory cells deep in the brain, we first sought to determine which areas of the hippocampus were important for memory encoding and retrieval, because there's a lot of different regions within the hippocampus even. So to do this, we test memory in a mouse. So how do we do that? We place mice in a chamber with a certain smell, a certain context, and we shock them 3 times. So this should become pretty fearful context for the mouse. 5 days later, we put them back in the same context and measure their freezing behavior. So freezing is a proxy for memory in this case. So if the mouse remembers the context, that it was scary, they should freeze, and they do, they kind of hunch up. And this is called contextual fear conditioning. And so if they don't remember, they'll be walking around. So if they have impaired memory, they won't be freezing. So we essentially designed an experiment where we tagged memory cells in a specific context, so call it context A here. And then we re-exposed half of the mice to context A and half of the mice to context B with different smell, different walls. So essentially, the mice should freeze in A and they shouldn't really freeze in B. And that's exactly what we saw. So we see an increase in freezing in A, not as much freezing in B. So then we wanted to look at the differences in the memory trace cells. And we found that in the DG and the CA3, there is a decrease in those memory traces in the B context. So we concluded that these regions were important for memory encoding and retrieval. So again, DG, CA3, we are showing that these 2 regions are important. Not necessarily, we don't know if they're necessary, but we know that they're important in memory encoding and retrieval at this point. So we then used a technique called optogenetics. I know that sounds crazy, a crazy term, but I'll break it down for you, to determine whether the DG or the CA3 was necessary for fear memory encoding. We saw that it was important, but we didn't know if it was absolutely necessary. So optogenetics is a biological technique that involves the use of light control, and it controls neurons and cells in the brain. So these neurons are genetically modified to express light-sensitive ion channels. And all that means is that these channels will respond to light stimulation, just a light flicker. And they're made actually from different types of algae. And so algae, if you don't know, actually move in response to light, and different bacteria as well. And so we harness the power of algae and bacteria, and we took some of their genetics, we threw them into the neurons so that these neurons would also respond to light. So optogenetics is basically a method that combines optics, light, and genetics to control individual neurons. And we can actually do this in freely moving animals. So we can implant the light right above the region that we want to stimulate in the brain, and then we can send pulses of light to those cells. So we can do this with a wire. Now they've become advanced, and we can do this remotely as well. So next we implanted the optoprobe, like I showed you, into the hippocampus, and we shined light on these cells that were labeled during the memory encoding. And we wanted to look at how this would impact memory retrieval. Could we actually manipulate memories? So we ran the same experiment, tagging in a context A, re-exposing the mice to context A and B. And what we found in the dentate, the DG, we turned on the light to inhibit neurons. We saw a decrease in freezing in context A. When we turned the light on, we inhibited it, but no difference in context B in both the dentate and the CA3. So when the light is on, we were able to decrease that memory of freezing in both the dentate and the CA3. Therefore, we concluded that the DG and the CA3 were active during memory encoding and necessary for behavior. And so I'll show you what that looks like in a video, because I know there's a lot of graphs here. So this is an animal here with optogenetic probes. And this mouse was tagged in a neutral context. So when the light turns on, the mouse should stop freezing, should start moving around, because we're manipulating that fear memory. So we're making them forget about their fearful context. So you can see the lights on, the mouse is more exploratory, moving around, no longer scared, because we have activated those neurons that were active during a safe context. So I'll play that again. So the mouse is freezing because it remembers this is scary. And then when we shine the light into the brain, we can make them forget. And it's not instant, but after a little bit of time, the mouse is moving around. So the next question is, what about engrams in disease states in which there is memory loss? Can we actually change this? And so there were some articles coming out of reawakening memory in mice that had Alzheimer's disease. So the question became, what if we could reawaken memories in people or in mice with Alzheimer's disease? And so Alzheimer's disease is something that I've been studying ever since grad school. It's a neurodegenerative memory disorder. And so in this disease, patients suffer mainly from memory loss, difficulty solving problems, a lot of confusion, depression, anxiety, a lot of different emotions, and difficulty recognizing other people. And this is a slow cognitive decline, a very sad disease. And so this is what's happening behaviorally. In the brain, we actually see neurons die, cell loss here. We see a buildup of really bad proteins that destroy cells, and these are called amyloid plaques. And so we were really interested, can we rescue these memories? Or are the cells dying? What's happening? What do the memory traces look like in an Alzheimer's disease model? So we bred an Alzheimer's mouse with our line that allows us to look at memories. And so again, don't worry about all the lettering for the mouse design. Just know that we now have an Alzheimer's mouse with our tagging line. And this is just an example here again of the hippocampus. The blue dots now are the plaques, so that bad protein that builds up in Alzheimer's disease. So this is just showing that these are the Alzheimer's mice now, but still with the tagging with the green and red cells. And so when we looked at freezing, we saw a decrease in the Alzheimer's mice, which we would expect, right? They can't remember the context because they have a memory impairment at 6 months of age. They're the black bars here, which also correlated to a decrease in that memory trace. So we saw the cognitive decline and we saw the decrease in memory traces, meaning they were having problems retrieving the memory, just like you see in Alzheimer's patients in humans. So we thought, what if we could activate these memory trace cells? Could we restore memory using optogenetics? So we implanted the optoprobes into the hippocampus. We tagged the memory just like before, same experiment in context A. And then when we put them back into context A, we turned on the light to see if we could restore the memory. And we were able to rescue memory in the Alzheimer's mice. So there are the blue dots here, the dark blue, and the control are in the light blue. So when we turn the light on, we are able to rescue that freezing and put it back up to control levels here. So we're essentially able to restore memory in mice that have cognitive decline by reactivating those memory trace cells. So it's very promising for Alzheimer's research. And a lot of people have moved on from this, and they've actually started testing this in humans. Instead of implanting optoprobes, which we can't do yet, they've used light flickering techniques with the same frequency. So placing a person in a room and flickering the light at a certain frequency, I think it's 60 hertz. And those patients are actually doing pretty well with recovering memories. So it does seem to be working, and it's very exciting. There's also techniques like deep brain stimulation that you can do in humans. They use this for depression currently, and they're also looking to use this stimulating cells using deep brain stimulation in Alzheimer's patients too. Okay, so those were the initial experiments. I'm gonna just talk about kind of what we're doing now, a couple future directions, and then I'll finish up. So a lot of fun, cool pictures and videos coming up. So now I've shown you the hippocampus. We've really manipulated that area of the brain. And so now we're looking whole brain. And I'm also very interested in sex differences. Alzheimer's disease actually affects women more than men, and I'm investigating why that is. So to look at memory cells in the entire brain, we created a pipeline very similar to before, and we label the memory in that context. We cut the brains, we stain them, we image them, and then we have this automatic cell counting pipeline, because you can imagine there are tons of cells in the brain, billions, and we have to find a quick way to quantify these. And so we've developed an automated system where we can segment the cells out of the brain. So we can tell a computer to figure out where the cells are, meaning they're darker than the background, or brighter, I should say, than the background. Then we can paste a mouse brain atlas on top of our brain slice. And so that allows us to pull out cell counts from every region of the brain automatically using a different algorithm, where we can then create brain-wide network maps for all regions of the brain, not just one. Okay, so this is an example. Let's see if the video starts. Of whole brain memory trace labeling. And this technique is called iDISC. There's a lot of fun names. So this is a picture of going through the mouse brain, looking at different cells. So you start at the front of the brain, and this is going through. This is an example of what I meant when I said we paste a mouse brain atlas on top of our slices. So all of these sections throughout are just different regions of the brain. And you can actually go, you can find these atlas online if you're curious and want to know all the parts of the brain. This is the Allen Brain Mouse Atlas. And there's websites for that are pretty interactive. And then, like I said, we can get the counts from each region. So these are all the cells in all the different regions. Sometimes we have to change the shape. Not all brain sizes are equal, so we have to merge them. And the final goal is creating this 3D visualization of memory traces in the hippocampus. So we hope to fill up this entire brain with cells as we continue counting. Okay, so labeling cells is basically a snapshot, right? That's a one-time picture that we get. It's not a live view. So what if we could record cells firing while the animal is performing a task? Turns out now we can. We can use a technique called calcium imaging, and it takes advantage of calcium indicators, so fluorescent molecules that respond to calcium ions by fluorescent properties. So you can essentially inject the neurons to have these calcium indicators, and they will fluoresce when they fire a signal. So you can implant a miniscope, shown here, to record these cells firing in real time. And so this is an example of calcium imaging. This is actually in an anxiety test. It's called the elevated plus maze. So you can see on the left here, you can actually see the cells firing in real time as the animal walks around throughout the maze. And these lines are just indicating when the cells are firing. So it's pretty exciting. We've shown that these firing calcium transients are higher when the animal is in a more anxious part of the maze, so the open arms, when there's no walls. And this is calcium imaging. We sped the video up here during that contextual fear conditioning task that I've been talking about throughout the talk. So you can see the cells. This is a little bit of a different contrast video, so it's easier to see the cells lighting up as the animal encodes this experience. So now we can get a snapshot if we want, and now we can also map that onto these neurons that are firing in real time. And we can look at how these change across days, across age, in disease states. All right, so I will try to sum up everything. I know that's a lot. If you guys have questions or want to learn more about anything, feel free to email me too if we don't have time today. But hopefully you've learned that Carl Lashley coined the term memory tracer engram. Patient HM led us to the importance of the hippocampus. Our lab created this activity-dependent tagging system to visualize memories throughout the brain in a mouse. The DG and CA3 are necessary and sufficient for fear memory using optogenetics. And Alzheimer's disease results in memory loss, a decrease in memory traces in the DG, but optogenetic manipulation can rescue this. And this is just another beautiful brain image. This is the ventral hippocampus, so further back in the brain. So with that, I thank you all.